Myself, Dr. Mriganka Mondol, would like to say about the anti-cancer activity of sphingolipids isolated from attenuated Leishmania pneumoniae. So, actually, what is attenuated Leishmania? Attenuated Leishmania is the one type of Leishmania that is parasite which produces the collagen. But in case of attenuation, due to attenuation, this parasite has no any effective disease productivity on the host. So, anti-cancer activity showed the lipid isolated from the attenuated Leishmania membrane lipid, and the cell death was also justified. So, multicellular organisms, we know that the homeostasis is maintained by regulating cell proliferation and cell death. From the early embryonic stage, cell division occurs in control area, but in mistake occurs in the cell cycle checkpoint. And what causes that? Causes the cluster of abnormal cell. Later, cluster of cells produces the beginning benign tumor, and then this benign tumor can easily remove through the surgery. But some cases, apoptosis does not take place, and what happens? The prolonged cell division takes place uncontrolled way. Some chemotherapeutic agents which are applied for cancer treatment induces the endogenous apoptotic mechanism for cancer cell death. Now, now I would like to say about the apoptosis versus the necrotic cell death. What is apoptosis? What is necrotic cell death? Apoptotic cell death means the gene regulated or the signaling pathway regulated cell death, that is DNA fragmentation takes place, decreases the cell volume, and the cell membrane bleeding takes place, and formation of apoptotic body is formed at the last of apoptosis. But in case of necrosis, the cell injury takes place and the cell membrane bleeding takes place and swelling down. Ultimately, the membrane breakdown and disintegration and inflammation occurs. Now, the lipid, which is a very important biomolecules, was isolated from the Leishmania. These lipids, actually the sphingolipids, are the complex family of the lipids. The basic structure is given in the figure, that is in sphingosine and fatty acid, and which usually the sphingosine fatty acids, large number of sphingolipids are made possibly by variation in sphingolipid backbone. Now, bioactive sphingolipids isolated from the attenuated Leishmania pneumoniae have a special interest for cancer therapy. Some chemotherapeutic agents create many pathophysiological problems, but in case of bioactive compound, um, which reduces the pathophysiological problems if we apply in the treatment of instead of the chemotherapy. So, sphingolipid destructions isolated from the parasites have special interest from cancer therapy. This sphingolipids was isolated from the actually Leishmania pneumoniae mastigor and induces apoptosis in both human cells and some mouse melanoma cells also. Now, objective of this work. Main objective of this work is isolation of sphingolipid destruction from attenuated Leishmania pneumoniae, then separation of major components of sphingolipid destruction. Its lipid fractions are collected and were named as LSPL1, LSPL2 and LSPL3. Next, justification of the anti-proliferative activity on several human cancer cells like HeLa, HG2, STML5, MIA-PACA2, HCT116, A375 and others melanoma cells. Verification of LSPL1 and LSPL3 synthesized cells, apoptosis through phase contrast microscopy, and malignant cells was also studied. Special attention was given by the apoptotic activity takes place by LSPL1 of two cells HeLa and K562. So, investigation of the detailed mechanism of apoptosis initiated by sphingolipids was done